And we're started. All right. I'll try to pick up where we left off. Just because of January and February, we get this all the time. Everybody knows this. Uh, First of all, I don't want, if it's not safe to go out, don't go out. I mean, that's why the virtual side is there. I'd be the same thing. I mean, if I got in the car this morning and it was just too treacherous on my roads, I'd turn around and come back and gone virtual. So I understand. It's not no problem if you want to contact me, contact me. But I kind of have to wait for the school to do its thing, and then we can adapt accordingly. That's there. So by and large, this was hardly a difficult day to get around, comparatively speaking. So far, who knows what it'll be over the rest of the weekend, but we'll see about that. All right. So much for that little bit of housekeeping. We'll start here. This is more or less where we left off, and the part I would like to get into, and I think it's coming up again. The sequencing of this particular, the way the PowerPoint in this text does it, is a little different than what I customarily do, but that's fine. I do want to get into historical figures, particularly. The history of diseases and microorganisms is particularly interesting. And then, starting next week, we'll get more into the nuts and bolts of the categories and classifications that are there. So, I mean, all of these things are, even we didn't know about them. Things were fermenting, before. So, bread and alcohol production, people they had grains and they were milling them, and all of a sudden they started to rise. They had no idea that there were yeast in the air. And the same thing with alcohol. The earliest alcoholic beverage, if you did not know, was mead. So they'd get honey from bees, okay, and they somehow extract that and basically just let it sit, and it would start to ferment. That was the original alcoholic beverage, and cheeses weren't a whole lot different. They used different things. They found that different plants had, by way of microorganisms, produced certain amounts of acidity, like an artichoke, and they would put that into a vat of milk that was warming up, of some type of milk that they obtained, and then magically they're cheese. Okay, just little things like that were interesting before we really knew about microorganisms and what they did and the concepts of fermentation. And you'll get sick and tired of fermentation from a laboratory perspective in micro, because we utilize that a tremendous amount. That's really the substance, as well as uh, staining and microscopy for magnification. So I'm guessing a lot of you had lab or have had lab already. It's mostly introductory stuff. And this is the microscope and all that good stuff, I would suspect. So other things, and yes, they use microbes, even elementary. So I mean, it's the old days when you used to use a variety of things that they would put on, not realizing that they were microbes, to undergo a concept with wounds and things called debridement or debridement, depending on how you want to pronounce it. And you'll hear about that term, particularly if you're in the nursing sphere. It's cleaning off dead tissue, and a lot of these, basically, what bacteria and yeast live on is a lot of dead stuff. They gobble that stuff up. Uh, I'm not as familiar with the precious metal obtaining, but this contamination is a big deal, and particularly so today. A lot of the organisms that we have tinkered with, and ones that are naturally occurring, are used to clear up oil spills and to be utilized in waste dumps and things like that. Maybe you're like we try to have sometimes, to have like a compost heap, because my wife will basically use that. Does a lot of organic gardening, and we throw all the stuff in there and make fertilizer, old cuttings and stuff like that. Just the same. Not maintaining that you have to, but a lot of people will utilize that. So that's some of it. And I touched on genetic engineering. Microbes are interesting. Bacteria, particularly. Some bacteria, and perhaps more often than not, most bacteria, basically, if you drew a bacteria and it's not doing anything to scale or anything like that, have a nucleus that's kind of super folded, and basically a big circle that's kind of folded in on itself. And a lot of times they'll have a little extra circle of DNA. So this is called the nucleoid. And you'll see it when we really do. And this little circle, I know you can't see this out of the virtual, but we'll get there. It's called the plasmid. And plasmid's a tiny circle of DNA with maybe around 20 to 30 to 50 genes. That kind of size is rather small. And what we can do is we can extract plasmids, and then we can take the plasmid and chop it with enzymes that come from bacteria. We wouldn't be able to do any of this if it wasn't for bacteria. They make very interesting enzymes that are, we talk about restriction enzymes, and you hear about that a little bit more as the program goes on. And so it can chop the DNA, and we can assemble DNA in the laboratory, a strand, and basically splice it in with similar enzymes. And then we can take it into bacteria that are sort of friendly toward absorbing. We call them recombinant DNA. So they'll absorb DNA in their environment with a variety of chemical maneuvering, and basically take this now modified plasmid and absorb it, and all of a sudden that'll reprogram the cell to make whatever we want. And the first genetically modified portion, maybe I told you, which I mentioned about insulin and give you the saga of that. Yes? No? No. The first genetically modified organ, basically organism or product that was customarily or used widespread was insulin. Okay. Insulin, if you didn't know that, we knew that insulin was something that could be obtained from animal pancreas and utilized in individuals to deal with their blood sugar. And if there's one disease, and I'll say this 1,000 times to all of you, that you need to know the most about if you're going to be in healthcare, it's diabetes. Okay, because you encounter more diabetic patients than just about anything else. It's a common denominator. So with insulin, what we utilize with that is it basically facilitates the absorbance of sugar into cells for an energy source. In the absence of that, a variety of whatever type of diabetes you have, and what's called type 1 is typically an absence of sufficient insulin. So when they started giving people insulin, they would grind it up from pork specimens, extract the insulin, which is a protein that was extractable, and they would inject it. And first of all, when you have animal products, you always have a lot of allergies because they're not human products. So a lot of people were allergic to it, couldn't tolerate it. A lot of people, for religious reasons, Islamic folks and Jewish folks wouldn't use it because it's a pork product. Okay, all those kinds of things were part of it. So it wasn't used. And they switched over to beef or bovine insulin. Same process. And it was a little bit better tolerated. Again, there were some folks who didn't want to use it. There were certainly allergies to it, which limited the treatment. And when molecular biology came along and we could analyze the sequence of this protein we call insulin, we could basically genetically engineer it in such a way to put it on a plasmid and insert it into E. coli or yeast, which are typically what we utilize for genetic engineering. And voila, we had humulin. Now it goes by a lot of different names, but the original name was human insulin or humulin. And we have long acting and slow acting. And there's a lot of stuff that many of you who go into those fields will learn about therapeutically. Why I use a certain type or why a certain type is prescribed. So that's really what it is, recombinant DNA technology. And we will go into that, and we do that in our genetics unit, which comes up probably in the second of the 4 units that we do. Bioremediation, again, is all about the pollutants, oil spills, biological waste, other kinds of waste. It's interesting. We make things using microorganisms because of the enzymes they produce that we don't necessarily produce or have trouble manufacturing. Okay, so the stuff that makes absorbable diapers, that came from microorganisms. That's the good news. Certain kinds of plastic products, we had to use that. But the trouble is they're very hard to decompose them. So we have to use other microorganisms to break them down. So it's good news because we can make useful products, but it's bad news because a lot of times the way to get rid of them from an environmental standpoint is difficult to obtain unless you use other microorganisms. There's a lot behind this that we use. And we use microorganisms, and we use technology that we'll talk about with regard to control of microorganisms, with regard to water filtration, things like that. See a lot of this stuff. So it's very interesting, I think. So that's the bioremediation portion of it. And this is, boy, they get a bad rap. A pathogen means disease causing. Gen means to generate or cause. Patho means disease. A pathologist is a doctor who studies disease. So pathogens, by definition in the microbial world, are ones that occupy us and thrive and in the process damage us. And we'll get to that definition down the road when we talk about things like epidemiology and host microbe interactions that are there. But there's only a couple of thousand that we're aware of. The big problem, and we'll get to it shortly, has to do with the fact that they're changing. The one thing about a lot of microbes is they're inherently genetically unstable and they mutate. And the best evidence of that is something called COVID-19, or killer strains of influenza. They are not the same as the original ones, and every so often a mutation occurs that we are incompatible with. And if you remember when COVID came out, if you listen to some of the talk on TV or in print media or other kinds of media, they talked about what's called a novel, NOVEL, virus. That means new and something we've never dealt with. And very often it's a confluence of viral particles and viral materials from multiple species. And that's where the whole idea of Wuhan and the lab and the open air markets and exactly how, and we still don't know for sure. Let's be clear about that. So, but infections are monstrous problems health-wise, much more so in some ways than they used to be, but not nearly as much historically. I mean, when you think about the plague decimating a third of the population of Europe back in medieval times, pre-Renaissance times, that's an immense number. Not on an absolute number of population, but on a percentage of people that died because of it. When you hear about, and the worst killer of all time was called the Spanish flu. That's influenza that occurred between 1918 and 1919 during that winter. And interestingly enough, it was an oddball. Usually when we talk about influenza type A, it affects the very young and the very old. This was one, and when we get to influenza, what it does is this one really provoked the immune systems of young folks like you. And because that occurred almost immediately after the end of World War I, where the armistice was signed on November 11th of 1918, that all of these young folks who had fought the war and survived, many of them died shortly thereafter. It was very, very tragic. It was we're talking about more than 100 million people. And I've read a lot about this. There's a wonderful book when we talk about flu that you can get probably fairly cheaply now because it's been around for a while. The lead health or science writer from the New York Times at the time, named Gina Kolata, capital Kolata, kind of wrote a book about that flu and sort of treated it like, it was almost like a biography, almost like a whodunit, like how it came about and all the things that they looked at. Very, very interesting. So infectious diseases are really significant. They have an interesting chart. They've changed it. So this country now, and the book is relatively new, whatever this is. So this is, let's say, within the last 4 years, statistically speaking, the biggest killers: heart disease, cancer. We'll buy that. That's pretty consistent. Heart disease, stroke here, less so with cancers here, and there's reasons for that. But heart disease, cancers, then accidents, and this is for here in the United States. Lower respiratory diseases, pneumonias of various types. That's the first of the infectious diseases. Okay, lower respiratory diseases. In this case, the context here, when we say infection and pneumonia, see where it's down here. I misspoke. Lower respiratory disease would be emphysema, chronic obstructive pulmonary disease from typically smoking related. So we have heart disease, cancer, accidents, environmental factors leading to respiratory disease, stroke, which goes along more with heart disease, cardiovascular disease, Alzheimer's, I don't know how accurate, diabetes, okay, mentioning before, major league problem. And all of a sudden now we finally get to influenza and pneumonia, infectious disease oriented disease causes, then kidney disease and suicides. This country. The distinction between worldwide and this country: availability and quality of healthcare, education, medical insurance, availability of antibiotics, lots of different things that we do. Heart disease and stroke, again, cardiovascular in nature worldwide. COPD, there's a lot more smoking and related problems with environment and leading to environmental respiratory disease worldwide. Now we're getting to the influenza and pneumonia, all the way up to number 4 on the hit parade. And then Alzheimer's, respiratory cancers, considerably lower. And a lot of that, I think, has a bit to do with the overall health and diet. That's a whole other issue that's there. Road injuries, okay, and then diarrheal diseases and tuberculosis. We'll get to tuberculosis shortly with regard, and we're going to revisit it when we talk about diseases, but sort of interesting because of the category it's in. We don't have as much problem. This is, and I'll say this a number of times, we have, by and large, it may not taste good, but, and you don't worry about going getting water. In the old days, we get it out of a water fountain, but now most of the time, the water fountains basically go into some kind of container. Because if you ever tested where the water came out, it's pretty gross. They learned about that. But understand, you're not worried about the water. If you get a glass of water wherever you reside, are you worried about it? Maybe it doesn't taste good. Maybe you're like us and we got a Brita or a Pure or a filter of some type, right? So it tastes better. That's the size of it. Get bottled water. You're not worried about it. There you got to worry. We have a, relatively speaking, pristine water supply. And waterborne and foodborne illness is a big, big problem. We just don't worry about it nearly as much. And you see it publicized periodically, and we'll talk more about that. When you see these epidemics and recalls and things like that, they're very, very significant. And most of the time they have to do with certain things that get pulled. And just every so often, look and see what's going on. See if something got recalled. Maybe it's something that you have in your house. Easily. So that gives you an idea. Emerging versus re-emerging, or what we call resurgent diseases. Emerging has a couple of reasons behind it that are pretty good. Emergent diseases, to me, the number one diseases we haven't seen is certainly, to me, mutation. Again, bacteria and viruses are very easily mutatable organisms. They don't have any of the safeguards comparatively that you and I do in our cells that preclude mutations. Our cells probably mutate on an average to about 1 in a billion, and we have a lot of safeguards to get rid of those bad cells before they cause us problems. Bacteria don't have those, and they mutate much more rapidly. We'll get into the numbers. Okay, areas we haven't gone to. So let's say building or development in other areas in remote regions, like going into a rainforest, going into an area that wasn't inhabited. I mean, what's one of the big problems we have around here, good old northwestern Pennsylvania? I live up in Cranberry Township. Any place that there were trees go away. Because they're building houses or they're building a strip mall or putting in a pharmacy, something like that. You've seen this picture. Okay, it's like, that was a beautiful area, and all of a sudden they're making houses that are, affordable housing from the 300,000 to 500,000. Maybe for you it's affordable, not necessarily for me. So we go in there, and what's our big problem? We're going into areas which are animal habitats. And particularly in Cranberry Township is the poster child for this, Lyme disease. So we didn't know a whole lot about Lyme disease, even though it exists, and I'll give you the litany about that down the road. But that's really a troublesome area because you've gone into areas where there's a lot of areas where ticks breed, and there's an abundance of deer around here. As you all know, there's an abundance of deer. I see one when I walk the dog most mornings. So building into areas. So if you think about in the rainforest, you might remember down, and there's a virus we'll talk about called Zika, when they started to have Olympic venues and World Cup venues in Brazil as they approached the Amazon and built in that area. And then all of a sudden they had a lot of people getting this virus called Zika, which is not fatal but causes some significant birth defects. So it's another one to sort of look at. That's there. So mutations going into these different areas, and scientifically, we're able to now identify things we couldn't identify before. So it's not so much that they weren't there, but now we can see them. We can find them. They have better technology to be able to isolate them. We can analyze their genetic strands. So it may be something, for instance, Lyme disease, when I studied about that before the advent of molecular biology, which is more or less around 1975 to 77. I matriculated in school in 1973, medical school. Basically, it was totally different. They talked about, I can remember them talking in my micro class, which is 74, tick paralysis. So we didn't even know about Lyme disease. We know that tick bites under certain circumstances could lead to forms of paralysis. The reality, it was probably a variant of Lyme disease. Now we can identify it a little bit better. So science and the identification, that's more or less what we begin to see about these things. And unfortunately, some of these emergent things, if you want to be a conspiracy theorist, and a lot of people are, you can think that they have been, biologically bioweaponized, which may very well have been the case with COVID. I'm not saying I have proof positive, I don't think anybody does, but there's a lot of suspicions, and it's like anything event that happens historically, you learn more about it 10, 20, 30 years later. They're still talking about the Kennedy assassination. That was 60 years ago. 60. So that's part of what happens with emergent. Re-emergent diseases, I call resurgent diseases. Kingdom for into like that. So re-emergent or resurgent means it's gone away, but it's back. Okay, two biggest ones, talked about it very, very briefly: tuberculosis, measles. That's right. Okay. Reasons follow a typical pattern. Vaccine laxity, worldwide travel. Real quick, because I should have commented about mutations, because I do a whole thing on that. Inappropriate, and this led to a lot of mutations, usage of antibiotics. That's more in the emergent. We do a whole unit on antibiotics. Antibiotics were misunderstood and mishandled, and still to this day sometimes. You have to be careful. But the resurgent diseases, vaccine laxity, worldwide travel. Okay, one of the things. The advent of immune disorders, HIV would certainly be in that, but it's not just immune disorders. It also has to do with their treatment. And basically, and that would be immunological suppressants. So, vaccine laxity, worldwide travel, immunological disorders, are all consistent with this. Immune disorders, and we've had a whole, just watch TV. How many people, if you have Crohn's disease, or you have this disease, or MS, or any of those things, you're seeing all of the, a ton of commercials, plaque psoriasis, other kinds of stuff. I take it immunological. So I have a variant of one of those immunological diseases. Take an injection every other week. Pretty simple, just like that. So there's a lot of different things. There's something else that goes along with it that plays a role, and I'm sure it'll come to me as we talk more about this. So nevertheless, where did the all come from? Tuberculosis. Start. Worldwide, it has always been a problem. The resurgence is much more so in this country. Tuberculosis has one common denominator: overcrowding. The country that has the greatest problem with tuberculosis, India, by far. India now is the most populous country in the world. It has overtaken China. I mean, we're talking about the better part of 2 billion people within India, most of them in the subcontinent area, the lower portion of India, not the upper portion as you're heading toward the Himalayas and stuff like that. Immense. If you go and read the population of the cities in India, it'll drive you crazy. The small ones have 9 or 10 million. Tuberculosis has been a big problem. Children there, if you want to travel to India, you have to be immunized by a very less than effective immunization that really works better on kids and doesn't work particularly well on adults. Okay? More about that story in a little bit. So there's an immunization that's required. If you go to travel there on just because you want to travel and see the Taj Mahal or look at the country, or you're doing a medical tour there or some research there. One of my advisees just graduated. She did a research project there in India, in the New Delhi region. Okay, it was for her honors project here. Just saying, there's a lot of that goes on with regard to it. They have tremendous problems with this because of that. It takes a long time. Tuberculosis is one of those interesting diseases that you have to be exposed to for a long period of time. When you folks get into hospital-based activity, you'll learn about what we call PPE, personal protective equipment. If you're assuming that it is enforced, and it needs to be, when you go into a TB patient's room, you have to have a special mask to prevent that. That's the kind of room where they have an exhaust pulling out, because when they cough or sneeze, which they do all the time, particularly cough, it pulls the TB out and spits it out. It pulls it into some type of a filter device so it doesn't go out. It's almost like reverse isolation in that sense. You're trying not to spread it to the rest of the hospital. Very, very interesting how it's done. We almost eradicated tuberculosis. How do we do it in this country? We never used the immunization because people would always test positive. Because you're giving them something that looks like tuberculosis. It's actually in the same bacterial family that's not as deadly, but should stimulate your immunity. I used it as it's often used in immunotherapy, which I had for my bladder cancer. You know. So all of those things are significant features that are there. So what happens in in this relationship, we used chest x-rays, treatment where it was necessary, and screening with the TB test that most of you have certainly had, right? Little subdermal test called delayed reaction hypersensitivity. If you're an AP2, you're going to learn about it with regard to immune system. And that worked tremendously efficiently. Then with the advent, particularly of HIV and age-related problems, and immunosuppressant drugs and transplants, and which I didn't put down there, transplants and transfusions, because those are all things that we see a lot more of today that tend to interact and compromise your immune system. We basically magically it came back. That's what's meant by resurgence. Measles vaccine laxity. Measles, people will talk a lot about vaccines, but it has a bit to do with some faulty research that people have seized on. And I'm not necessarily an advocate for vaccines. I certainly would not have been an advocate for this recent COVID vaccine or the 63 others they keep giving people. Okay, because they haven't got the first one wasn't right. I'm not an advocate whatsoever for that. But the reality is that there are good vaccines and there are bad vaccines. The MMR is a great vaccine. Had minimal problems. All of you had that. Measles, mumps, and rubella. Here it goes. When I was a kid, we didn't have any of those. That vaccine didn't show up until 1980s, maybe pushing 1985, 1990, that kind of a thing. The chickenpox vaccine didn't show up till like 1995. All three of my kids had chickenpox. In my day and age, we had what they were called the usual childhood diseases. I had three of the four. I had measles, mumps, and German measles. Okay, I beg your pardon. I had measles, mumps, and chickenpox. I got an immunization when it finally was available. It was about 1980, 1978, 79, 80 for mumps, and it was relatively new. We didn't even have it combined with MMR. But people, because of have become, and a lot of times has to do with state laws, a lot of times has to do philosophically or religiously, and they have become more lax. And what happens when you get a lax vaccinated population and they go into a populated area, measles is still around. It's the third largest killer in sub-Saharan Africa. It didn't go away magically. You just don't hear about it. They had an outbreak at Disneyland because a group came that was not vaccinated, and they had active measles, unfortunately, and there were other unvaccinated people. And you go to one of those theme parks, look at those lines, right? You're wall-to-wall people for half an hour, 45. You're going to some popular ride, 2 hours standing in line. Just giving you an idea. So that's kind of little information that you should be aware of. And if there's any more causative factors that I don't recall off the top of my head, I'll make sure I give them to you. I might even have them in the handout. If I have them in this, I might have them in this handout. Hold on. So I'm forgetting stuff these days. Watch out. I say this to all classes. If you get contacted from an Adam M. Stein in the Navy, is my dad doing anything we can put him into a home for? Let me know. Just be aware. He's convinced. I'm this close to walking around without clothes. He's just totally convinced. You may have heard me, and you'll hear me say it again. Okay. Resurgence due to, okay, travel, vaccine. The one I forgot. Aging. We're living longer. That, you know why I forgot that one. I don't want to admit it. It's an opportunity for you heard all these. You guys, this crew over here, they've heard all these bad jokes. Admit it.
They're good jokes.
God bless you.
They're hilarious.
God
I
bless
cackle
you.
every time.
Yeah, cackle is the right word.
I know.
For those of you not laughing,
Oh.
Spaceballs, the movie.
Okay.
Just saying. Aging. No wonder I forgot that. We call that a Freudian slip. Okay, so, and we'll talk about these. Like you see, these are things that weren't there. And a lot of it, AIDS is an interesting story. When we get to viruses, we'll talk about it. Hep C probably is a mutation. Zika, West Nile, again, those are viruses that probably came from going into areas where we identified or released or became acquainted with things. And TB, certainly, again, the poster child for researching. For those that might have had me and we discussed this, gastric ulcers are not necessarily caused by health. They could say associated, I'd buy it. If they say gastric carcinoma, I would buy it, gastric cancer. I have had 2 major surgeries for gastric ulcers, one in 1993 and one in 2000. I never tested positive for that. So, I mean, is there an association? I'll buy it. These other things, okay, MS, we'll talk about a herpes virus that has a strong association, not necessarily a cause, but a statistical association. Obsessive compulsive disorders, there's speculation. Heart disease, certainly you can make a case for certain kinds of microorganisms, even certain kinds of obesity. There's a lot, we're learning more about this stuff. It's not cause and effect necessarily. It's like, for instance, there's a form of Alzheimer's that most of us would grant you are associated with prions, a very small percentage of known Alzheimer's, one particular substrain, with these, because it mirrors what goes on with these misfolded proteins. And we do that in the viral unit. So the weakened defenses, really a big topic. It's in any environment, We talk about it ecologically here and environmentally. We don't dig for something. Okay, we're protecting the environment, but now we have to get other sources to supplement for that. So you can't do one thing or not do one thing without impacting it. The same thing happens here. We come up with these wonderful, what they call biologicals, like the one I'm taking, that fights a particularly called pro-inflammatory agent, something that leads to inflammatory and starts to block it in your bloodstream, that people with certain diseases have. The trouble is, it weakens you. The saga of my bladder cancer, which was discovered about 8 years ago, it was relatively tame. They gave me this immunological agent, immunotherapy, the same drug that they use for as the tuberculosis immunization to bolster your immunity. It, as every time I was exposed to the drug, I had more excessive reaction immunologically. I was really in bad shape the day I got it for several hours, and it got progressively worse. Then all of a sudden, I'm starting to get tremendous amount of pain and swelling in joints. It probably led to, at the very least, initiated my exposure to that. And so now, and then I end up on immunosuppressants. So I was doing, by bolstering my immune system, it created an immunological disease, and now I suppress the immune system. And unfortunately, that might make me more at risk for other things, which is why, other than eating excessively, I try to stay relatively healthy. Don't start.
I feel that.
Don't start. It's all right. So that's why my wife frequently tells me to get my corpulent posterior to the gym, because she wouldn't say fat ass to the gym.
No, she wouldn't. Queen. She is queen.
Mm-hmm. Tell me about it. I said, she can't talk back. She uses that tone with me. I've told you how I respond. Yes, dear. Right away. That's why I'm here talking to you. Okay. Drug resistance. That's the antibiotic thing. We're going to get into that. Yeah. They may have. I put may. I hate absolutes. That or that. Okay. So we're going to do a whole unit on the difference between membrane-bound organelles and eukarya, and go over nucleus and mitochondria, etc. Okay, and versus bacteria, archaea, which are remarkably smaller, they have organelles, not membrane-bound organelles. Again, it's a PowerPoint. Don't trust it. That are microorganisms, and by definition, all bacteria and archaea are microorganisms. They're single-celled entities. Eukarya may, not always, be macroscopic. Viruses, acellular forms. They're not made out of cell. There's somewhere, depending on the level of complexity, between large molecules and cells. And they have, and this is the key feature, I'll say it a bunch of times, DNA or RNA, never both. They always have protein coat. They may or may not have a membrane. I'm going to reiterate this again, and I have a handout for it that I have online. And prions are simpler, yes, because they're only proteins. He give you some interesting pictures. It's more lab-oriented. Again, when you're talking about a worm, it's like identical segments. That's why I don't do a whole lot with them. Fungi, the way we identify them is how they reproduce, and they sporulate. They spit out these spores. And in the lab, you may or may not, depending on what your instructor wants to do, and we typically look at pre-made slides of those. Protozoans, and you probably saw this in pond water. It's one that has like a stem that seems to attach, called Vorticella, and it has like little cilia, and it's sort of, and the cilia move the water, and its little particles come down, they feed on it. Good old E. coli, it's a bad slide, you'll see much better ones. Herpes is a viral particle. It has, interestingly enough, it's made of DNA. And prions, you can't really even tell that those are folded proteins. You'll see better pictures of that. We're not going to do the largest and the smallest. We're not going to make a line of development. We're going to talk about history. I love it. So in the time we have left, which is not a lot, I'll introduce the historical aspect of it. All right. I was hoping to get more of it. The whole idea was spontaneous generation. It's like you'd see in a sci-fi movie that somebody, the wizard or the witch, whatever, with the sorcerer would do their magic wand, and all of a sudden the table would start to move, a dead thing, a non-existing, and basically life out of lifelessness is what spontaneous generation meant. Another name, so they called it abiogenesis. Bio for life, genesis for making it, a for without. Life from lifelessness. We know that that's not something that happens. Okay? So even after the microscope was developed and we started to see particles, they didn't believe that. So the theory was called spontaneous generation. And much of the history of microbiology started with the disproving of this theory. And certainly this is mostly about Pasteur. Perhaps the most important, not the only one. Okay. So living things only arise from other living things, unless there's and again, they didn't know anything about viruses or prions. So Pasteur postulated, in other words, he said there's something there that we can't see with a microscope, and he called it a virus. He actually named it. Virus meant poison. He did not know that this was some interesting particle that didn't meet the definition of life, but he did coin the term. And Pasteur will be the most famous, and we'll get back to that, and we'll look at the swan neck experiment. Microscopes started with Hooke and von Leeuwenhoek. You probably got a little bit of this in lab, maybe you didn't. Okay. Hooke fashioned the first compound microscope. It meant two lenses. When you compound them, you multiply. Like when you're looking at your microscope in the lab, you got 10 for the ocular, and let's say 10 for the low dry. Boom, now you got 100 magnification. Okay. And he sketched them. He looked at plantings, cork, and things like that, tree bark, where von Leeuwenhoek was not a scientist at all. Hooke was. Von Leeuwenhoek was a fabric merchant. And when he would travel, he wanted to have something where he could study the fabrics as they were made, the thread counts and things like that, to see what their value was. And when he traveled and he was done looking at that, he fashioned a little single lens, which a lot of schools used to make, and their science labs just for fun. And he observed what he called animalcules. Okay. But, from teeth. And I used to do that when I did the lab, teeth scraping, so you could see all the stuff that was in your mouth. Okay. And constructed all these ones, and they actually had a magnification about 300. And historically, in a lot of PowerPoints, you'll see what this looked like. And then we'll talk about Kohn, we'll talk about Semmelweis, we'll talk about Lister, Pasteur, and Koch. They're all part of it. So I'll take you earlier, and I have this handout on Blackboard, historical figures. And I don't know whether they're going to have it in this PowerPoint per se or not, but the first one of any note was in the 1600s. His name was Redi. He was Francisco Redi, who was an Italian biologist. And he made an observation. When people left meat sitting out on a plate, as soon as flies started to land on the meat, shortly thereafter, he started to see maggots, tiny worms start to form. Okay. We don't do that. We either put it in the fridge, we have it in a zip top bag or whatever, right? All of those things. So he said, I think that those flies are come and laying eggs, and that's what's producing the maggots. He was absolutely right. And he constructed an experiment with 3 dishes. One, and it was like an old style petri dish like you'll use in the lab, except it's made out of glass. He put 3 identical pieces of meat. He put a glass top on one. No air could get to it. Didn't spoil. No evidence of maggots. He left one open, maggots, and he covered the other one with fine mesh gauze, what they used there in Italy, cheesecloth, that they still do to make cheese. You put the curds in there and the water comes. Anybody ever make cheese with cheesecloth at home? Wow. Paneer. It's Indian farmer cheese from India. And what happened was the mesh gauze was so fine that the egg, even though the flies lit on it, the eggs, microscopic eggs, didn't get through. And people were battling, scientists were battling about that because they couldn't duplicate it. When you validate something scientifically, somebody, if you want to prove it or disprove it or substantiate it, you have to use the identical things and see if you get identical results. They didn't all have the same cheesecloth. So there was a lot of controversy over that. That was in the 1600s. And then maybe the most important, and I think we'll conclude with this, was Edward Jenner, around 1791. Are you familiar with Dr. Edward Jenner? Aha! One of the worst diseases of mankind, and effectively his work eliminated it. We're talking about smallpox. In addition to plague, the other giant killer in the Middle Ages, smallpox. The ultimate bioweapon, a respiratory disease that's a virus that kills in about 3 days. And all you need is somebody here coughing with smallpox, and a bunch of us are going to get sick of it. He made an observation, scientific theory 101. He observed patients with smallpox when he saw milkmaids, and they didn't have anything automated, 1790s, they got cowpox on their hands. But if they got the cowpox, they never got smallpox. He reasoned that there was something in the pox. The pox is a pustule. So he took fluid out of the pox, and whatever he did with it, his idea was to inject it into people and expose them to a room with smallpox, which is what he did. You always test it on yourself, your family, people who are willing, etc. That's great. We still do stuff like that. We get prisoners to volunteer and students to volunteer for testing all the time. It worked. If it had been wrong, I'm sure he would have been executed because he would have been viewed as a murderer. How can you give somebody a shot from stuff from a cow, stick them in a room with smallpox, and if they died, it's on you for sure. You're a murderer. Well, that wasn't the case at all. And he developed the first immunization. And what do we call immunization? Vaccines, right? What's the Latin word root for vacca? For cows. So he derived the word vaccine, came from the Latin word root vacca, for cows. So started with that, and that was the first immunization that was used. This is roughly 1791. By less than 200 years later, we have not seen a case of smallpox since 1977 in Somalia. We used to all get I got it. You didn't. You used to all get immunized for smallpox. We have a very much older relative. It was such an oddball that ladies particularly would get these big scars from where they injected it. It had some type of local reaction. And so if they had a particular, like an arm that got a little flabby, all of a sudden you said this giant circular thing. I remember on older relatives. My mom had one that was pretty distinctive, but not that large. So it's interesting. We will continue with our discussion of historical figures and get to Semmelweis and Lister, and then finally to Pasteur. Thank you.